Hello, and welcome back to this course in which you will learn the fundamentals of artificial intelligence, gain vast exposure to topics such as Python, gene expression tools, and more. In today's video, we are going to be diving into the field of bioinformatics, which is truly one of those fields which has intersections between biology and technology, and it's really a mix of multiple different professions, so it's very important to understand. Specifically, we're going to be looking at Gene Expression Omnibus, which is a key resource when it comes to gene expression data, and we will use it to find our dataset. This video is a follow-along video, so if you have your own laptop or device, please follow along as we go through the steps. All right, let's get started. All right, so as I mentioned, we are going to be using the Gene Expression Omnibus tool as our main data repository throughout this project. But what exactly is Gene Expression Omnibus? Let's take a look. So if you open up your web browser and search up Gene Expression Omnibus, the first link that shows up is actually the link that takes you right to the platform. So now what's important to understand about Gene Expression Omnibus, the repository, is that it's just that. It's a repository of information regarding gene expression and gene data analysis that other scientists have been able to publish their materials and their results to. Now, this program is brought to us by the NCBI, or National Center for Biotechnology Information. It happens to be an organization which supports many of the projects that scientists are doing today, and their support is super important for tools such as the Gene Expression Omnibus. Now, another reason why the Omnibus is super important towards scientific discovery as a whole is that because it makes data super accessible to anyone. You can almost compare it to a super successful software company open sourcing their code, right? Now, this provides people with the unique ability to access research data and gene data without ever having to set foot in a lab, which is obviously super helpful in any situation, especially during a pandemic, right? Normally, gene expression data is one of the harder data sciences to do, especially during inside a lab. So the fact that we're able to access millions upon millions of studies, tens of thousands of data sets, right, as you can see here, is really, really helpful. Okay, so now let's walk through the different parts of this tool and actually find our data set. All right, so to get started, we can just head over to our search bar right here and type in multiple sclerosis. Now, keep in mind that Gene Expression Omnibus as a whole and this project as a whole are really just guidelines for any sort of disease. So if you wanted to search up about Parkinson's, or if you wanted to search up Alzheimer's, or if you wanted to search up any real disease today, I can guarantee you that some sort of genetic material and some sort of genetic study is available for that disease. Gene Expression Omnibus, like I mentioned before, is just a repository of thousands upon thousands of data sets, as we can see right here, series, platforms, samples, all of which have been accumulated by previous studies. So it's all available for you, and what we're teaching you in this course is just a format and a guideline for you to work this out on your own, maybe with a different disease if you so choose. But given the fact that our project is centered around multiple sclerosis, let's type in multiple sclerosis in the search bar and get started. So once you click search, actually two links will come up, right? You have 5,208 results for multiple sclerosis, and then this much larger number for multiple sclerosis as well. And what Geo or Gene Expression Omnibus does is it just filters down the results from this really big number to this really small number. So what you're going to want to do is click the smaller number, and it will redirect you to this screen. Now, I totally understand that this screen may look a little bit intimidating at first, right? You have a bunch of links, you have a bunch of text, a bunch of stuff all around here, right? Like these search details, it's kind of confusing. What does GDS mean? What does this database selection mean? These filters, all of these sort of filters on the side, all of it is just a little bit confusing. But what you need to understand is you don't need to understand each and every line of the stuff that is covered here. We are going to be explaining to you just the bare minimum that you need to understand, but I highly recommend that you search up on YouTube how to manage through geo datasets, as it is really cool. And if you want to use it for future science fair projects or any sort of projects, it is a really, really great source. But for now, we are just going to be clicking on the dataset. So click datasets. Now this filters it down to just datasets. So this, I think, is a good time to really explain what we're looking for. So once you've managed to filter it down from datasets, right? Once you click on that datasets, now what we want to look at are these two top organisms on the side. So normally, right, before we click datasets, there were a bunch of different organisms right here, all written in their Latin scientific names, right? And it can get a little bit confusing. But actually, Geo has this incredible tool where if you just hover over the organism, it'll give you the non-scientific name for it. So for example, Rattus norvegicus, I probably butchered that, but if you hover over it, it just tells you, oh, it's rodents. So you sort of understand that 14 data sets are available when it comes to multiple sclerosis, and your organism is rats. If we want to go to, so rodents, right? Now, we should know that Homo sapiens are primates, which are humans. So the fact that we have human genetic information when it comes to multiple sclerosis widely available to us is absolutely crazy. But we're going to be using Homo sapiens. So click on that data sets again. And as you can see here, we only have 13 studies, 13 data sets, I should say, when it comes to multiple sclerosis in humans. So we can click that little 13 and right next to it. It usually takes a second. But once you manage to click it, you can then see that the total items right now are 13. So what we've done so far is we've filtered down all of those 5,000 search results that came to us originally, and we filtered it down to multiple sclerosis and genetic data sets in humans when it comes to multiple sclerosis. So now let's sort of take a pause and see that we are getting a bunch of different data sets, right? 13. And they're all a little bit confusing, right? We have, for example, analysis of PBMCs from patients with clinically isolated syndrome suggestive of multiple sclerosis. We have multiple sclerosis brain lesions and analysis of histologically characterized multiple sclerosis. So there's a lot of stuff here, right? And we're looking at multiple sclerosis in patients with high serum of a certain drug, right? So there's a lot of data sets here. But what exactly are we looking for in our project? Our project is trying to compare the genetic expression of certain genes in a normal control level versus a person with multiple sclerosis. But before we go too in detail about what we're looking for, let's just discuss some of the qualities that makes up a good data set and what you would be selecting in a Geo data set if you were doing this project on your own. So number one needs to be the amount of samples. Keep in mind that these samples are comparisons between controls and between the number of multiple sclerosis patients. So normally, right, if you have something like 7 samples, that only means that they have 7 genetic outlines of 7 different patients, which would be okay if you're using maybe a smaller project, right? But given the fact that we're doing an ML and AI project, we really need a lot of data sets. So something like 29 samples would be much better than something with, say, 7, because we need to train our model, we need to get all that training data, testing data, all of that. So normally, if you're doing any sort of project on your side, you really need to identify what is the level of samples that you need. Okay, in addition to that, we also want to make sure that these samples have a control, right? We want to compare our MS gene expression data with something that is normal, otherwise known as a control. So it's very important that when you're selecting these sort of data sets, you want to make sure that there's a lot of samples, right? So probably not 4, but a lot of samples and a control. All right, so now let's go a little bit more into detail and really identify which sort of data set we want for our project. We've already identified that we want something with a high amount of samples, and we want something with a lot of samples with which compares between a control and a diseased person. Now, number 2, one of the major things that we want to talk about is that our research is not specific to one type of drug. Most of these data sets, as I'll go into a little bit more specifics a little bit later, but most of these data sets that are given here are seeing the effect of a specific drug that perhaps a team of scientists have developed, and they've injected it into someone, and they want to see the effect of that drug, right, in a patient. For example, number 1 right here, right, they have this extremely interesting looking drug, in vivo treatment effect of CIS multiple sclerosis patients, right? So they're comparing the different levels of multiple sclerosis and gene expression in a patient once they've been subjected to this treatment that they are thinking will help. That is not what we want to look for. Given the fact that we are trying to develop a neural network that can diagnose or predict early onset of multiple sclerosis, we want just pure, simple genetic expression from a control versus people with MS. No fancy drugs in the way, no fancy, treatments on the side. We're just looking for plain, simple MS versus control. Now, another thing we want to look for is peripheral blood mononuclear cells. Now, what exactly is peripheral blood? I've been sort of scrolling around, and we see that a lot, right? We have peripheral blood mononuclear cells. We have peripheral blood mononuclear cells, peripheral blood cells, peripheral blood mononuclear cells. So as you can sort of see, we're seeing those same words a lot. So what exactly does that mean? Let's go back to the drawing board. All right, so when you search up mononuclear cells, this is the definition that comes up. Mononuclear cells refer to blood cells that have a single round nucleus, such as lymphocytes or monocytes. When isolated from circulating blood, they are called peripheral blood mononuclear cells. But other sources exist, such as the umbilical cord, spleen, and bone marrow. Okay, great. So we have established here that mononuclear cells are just cells which have a single round nucleus. But you might be wondering, well, don't all cells have a single round nucleus? Well, no. Actually, when you think of a blood cell, right, this is sort of the typical shape that comes to mind, right? This sort of flat, round cell with that little cavity inside, right? You have a bunch of these sort of cavity-filled red blood cells floating around in your veins and arteries, right? A bunch of them. But what exactly is that cavity? Normally, when cells are really white not white, but filled up, right? That means that they have a nucleus. Red blood cells actually don't have a nucleus, which therefore means that they don't have genetic information. But this is where mononuclear cells come in. These mononuclear cells, like white blood cells, actually do contain genetic information. Now, what is special about white blood cells? White blood cells play a very important role in your immune system. Now, you can sort of draw these connections yourself, right? We have white blood cells, which is important to the immune system. And we know that the immune system is important to multiple sclerosis. So you can draw that connection between multiple sclerosis and white blood cells. Otherwise, in this case, known as peripheral blood mononuclear cells. Now, the peripheral part just comes from when isolated from circulating blood. That's all it means. Basically, when the blood is still inside the veins, it is just regular blood. When it's taken out of that, it is known as peripheral blood mononuclear cells. So, long story short, all you need to know is that peripheral blood mononuclear cells, in this case, refers to immune cells inside the blood, which showcases to us that we are on the right track. That is what we want to see, because we are comparing genetic information in multiple sclerosis, which checks all the boxes, right? It's immune, genetic information, which comes from those single round nuclei, and it is in the blood. So all three of those things are showing up, and that is what we want to see. So given this data, given all of these things that we have seen, let's go back to our datasets and see if we can identify one sort of gene dataset that allows us to use all three of these things and see what genetic information differs from people with MS, the genetic information that's in their diseased blood, versus people in a control sample which have regular blood. Let's go back to the gene expression omnibus. All right, so we are back in our gene expression omnibus, and we are finally on our way to decide our very own dataset that we can use for our project. So let's just go one by one. There's only 13, so we don't really need to, just look at it from a broad point of view. But normally when you're doing a much bigger project or a much more common project, there will be a bunch more search results, right? You might have, if you're doing Parkinson's or something, you might have maybe 130. So going one by one is not the way to go. But for now, let's just go through it so you get a better understanding of how we want to find our data set. So number one, we already talked about this, right? They're talking about a drug effect, so we can just skip that one because we don't want to see a drug effect. Number two, this is actually not bad, right? They're looking at brain lesions in multiple sclerosis, but unfortunately, it does not have nearly enough samples that we want, and it's also not talking about blood. Moving on, we have analysis of peripheral blood mononuclear immune cells from multiple sclerosis, so that's good. In MS patients and unaffected controls, so another check mark, MS patients versus controls, and it just talks about results provide insight into the molecular mechanisms of underlying MS etiology. Awesome, and it has 29 samples. Great. So all the boxes click on this one, so we can just open that up in a new tab. Whoops. Let's open that up in a new tab and save that so we can come back to it. Moving on, we have MS, patients with a high serum. Oh, so this is, again, a different type of drug, right? So we can skip that. Again, whenever it says something like effect, so like some weird sort of looking drug effect on MS, right? We can just skip it right away. Oh, this is interesting, right? This is a same one where it's talking about PBMMCs from multiple sclerosis, excuse me, and it has 12 samples. So it looks exactly the same as this other one over here. So we'll open that up a little bit. We'll open that one as well, just so we can take a look at them. Identical twins. So this is actually pretty interesting. They're looking at specific cells though, so we don't really want that. We'll skip that. Again, this is a type of drug, so we'll skip that again. And yeah, the rest of these are just drugs or with very little samples. So I think we have our two that we want to go with. But now this is interesting. Let's take a look at them. Number 1, analysis of peripheral blood mononuclear immune cells from multiple sclerosis patients and unaffected controls. MS is an inflammatory neurodegenerative disease. Results provide insight into the molecular mechanisms underlying MS etiology. Okay, so it sounds very good. It sounds like it has everything we need, but there's another one right here with a slightly less sample count, but if it has something that the other one doesn't, we should choose this. And as you can see, it is the exact same summary. So in cases like this, and there are bound to be cases like this where it seems like there are two data sets with the exact same sort of information, NCBI and Omnibus have to have a citation, right? That's where they got this data from. And what's great about this is it has a link to the exact article where they published their data, where these researchers, I'm not going to try and pronounce their names, but these researchers have actually released their study. So if we click on this link and open it in a new tab, we can actually see their paper that they wrote about that gene expression data that they have submitted to the Omnibus. So if we do that for both, right, this is by these group of scientists right here. So if we open up their data set as well, you don't need to follow along for this part since I'm just doing it right here and you can take a look. But now we have two different studies from two different data sets that look identical. But if we take a closer look at the actual study that they released, we can see it's anything but similar. Number 1, right, with a 29 sample, systematic review of genome-wide expression studies in multiple sclerosis. So long story short, they are looking at genome expression in multiple sclerosis, which checks our boxes, very good. Now the second one, role of HDAC3 on p53 expression and apoptosis in T cells of patients with multiple sclerosis. Okay, this sounds cool, but it's talking about a role of a certain gene or a role of a certain level of expression or a role of a certain little thing on multiple sclerosis, which really isn't what we're looking for. We're looking at a systematic review of gene-wide expression, right? We want a broad gene expression view of multiple sclerosis. So that we can sort of eliminate the second geo data set that we have right here. And we only did that by actually looking a little bit further and looking at the actual study that was published. So great, we actually have our very own geo data set with us right now that we're going to be using for this project. 29 sample counts, and we have our data set. Now, if you go back to the geo home screen where we actually got that data set, what you want to do is instead of clicking on the link, which I did before, let's just go back there. So instead of clicking on the title like this, right, which leads you to the summary page, you want to click on this, where it says series, and it says GSE21942. It might be a little bit different for you, but you want to click on this series. Now, once you click on this, this takes you to the Geo Omnibus tool. Now, when you see here, you can sort of see these more. And this is where you go. This is exactly what we want, right? We have a bunch of controls. We have 15 controls, and then we have 12 with a few replicate MS patients. And this is exactly what we want, right? We have a bunch of genetic information, genetic level of expression information about controls and MS patients. In the next few upcoming videos, we're going to be taking a deeper dive into the Analyze with Geo2R tool, where we will actually go into seeing what sort of types of data we can accomplish and we can extract from these data sets, and we're going to be learning about how we can sort of see what data is actually hidden inside these many rows. So with that, there's a lot of fun stuff ahead, and let's go to our summary. All right, so we have seen a lot about bioinformatics and the bioinformatics tool Gene Expression Omnibus today. Let's break down what exactly we saw. Number one, we were introduced to the tool and how to navigate through the various levels of datasets and large number of projects on the tool itself. We really saw what exactly Gene Expression Omnibus was and how to really use it to our advantage. And we learned about all the various pros and some cons about it. Now, most importantly, we were finally able to identify the gene dataset that we are going to be using for our project, which is a huge step towards our project. We learned about the criteria we needed to follow when deciding which dataset to look for, including sample size and making sure that the study was relevant towards our goals and all of that. Now, that concludes the second video of this series. You should be very proud of yourself for watching this. In the upcoming videos, we are going to be taking a much closer look at what exactly I mean when I say genetic information. I've been talking a lot about, these studies containing genetic information, etc. But what exactly do I mean when I say that? We're going to be diving into that. And we are actually going to be manipulating the data we have to make it usable for a machine learning program. So we are actually going to be utilizing bioinformatics, and we are going to be little data scientists ourselves in the upcoming videos. So a lot of fun stuff ahead, and see you in the next video.